Dear participants of BIMCO 2023, you are now at one of the events of the Congress, namely at one of the lectures. And it's a great pleasure for me to introduce to you our speaker, Professor Tiago OUTEIRO, Professor of Aggregopathies, Director of the Department of Neurodegeneration and Restorative Research of the University Medical Center, Gottingen, who is going to talk to us today about molecular determinants of synucleinopathies, insight into therapeutic opportunities. In case any questions occur, please contact either our OC or professor directly. We would also like to remind you about the basics of copyright and intellectual property. The use of any material from the lecture must be done only with the consent of the lecturer. Thank you and enjoy the lecture. Professor, the floor is yours.
So, hi everyone. It's a great pleasure for me to be with you here, just online. I cannot even imagine what you're all going through. And I would have loved to be there with you in person. I know it's still not possible. I can just hope that this will end as quickly as possible and without any major losses for any of you and your family members. I believe most of the world is with you during this difficult time. And I really hope that we can overcome this and have many in-person BIMCO meetings in the future as you deserve. So I would like to say from the beginning that if any of you is interested in research and if there's anything we can do to help, feel free to get in touch at the end, and we'll try to help and do whatever we can to support you. So
thank you, Professor. I would like to thank you for such kind words. Yeah, it's a big pleasure that you could join us this year, despite of the problems that we have. We are very happy to have you here today with us.
Yeah, it's a pleasure and an honor for me. So really, I meant every word I said. So now to the science. I will talk to you about some work we've been doing in my group in the context of neurodegenerative diseases. And in particular, I will focus in one group of these diseases that we call synucleinopathies. And I will get more into detail so you know which diseases I'm talking about. So I'm talking to you from Germany, from Gottingen. I'm not German, as you may have guessed from my name. I'm actually from Portugal, so even further away from Ukraine. But I work in Germany. I work in this city called Gottingen, which is in this region of the country you can see here. And this is our website, so you can see more about the work we do in our website. Göttingen is a small university town. It has a big university, and most of the businesses here are around the university. These are some pictures of the city, so you're all invited to come and visit in the future if you want. This is a very well-known city for many famous researchers that passed through Gottingen in the past. So this is an observatory where Gauss, the famous mathematician, would study the sky using his telescope. And for such a small city of about 100,000 people, it's quite impressive to think that more than 40 Nobel Prize winners were in Gottingen at some point, so doing their work. And so it's quite impressive. I'm very honored to be in Gottingen as well. But what I want to tell you about today has to do with this idea that protein structure is deeply connected to the function these proteins perform. So here you see three examples of very different proteins that fold into very different three-dimensional structures. Some of them occur inside cells, some occur outside cells, and they all have a function to perform. And the function depends on the structure. So if we change the structure of the protein, we will most likely affect the function as well. And a process that is represented simply here that we refer to as protein misfolding is this process from where a protein that normally has a determined three-dimensional shape changes its structure. And then we know that at some point protein aggregation can occur. And as I told you, if we change the structure, then we change the function. So when we have protein aggregates forming, then we lose the normal function. But we also talk about the gain of toxic function. So it seems that some of these protein aggregates can actually become toxic. And we know both of these can contribute to disease. And this is true in a variety of different diseases. So this process, again, represented here in a simple manner. Here, of course, this is induced by temperature. When you fry an egg, then you denature, you misfold the proteins in the egg. And the proteins are still there, but they have different biochemical and biophysical properties. And this process is related to cancer, to diabetes, to neurodegeneration, and maybe even possibly related to the COVID-19 pandemic. When we talk about neurodegenerative diseases, these are the main diseases we usually talk about. Of course, the most common of these diseases is Alzheimer's disease, followed by Parkinson's disease. And then there's several other diseases that are less common, but equally devastating. So all of these diseases that affect the brain are devastating. They cause severe problems. And you as future medical doctors will deal, unfortunately, with a lot of these during your careers. What we know is that there's something common among all of these, and this is the accumulation of misfolded proteins, of aggregated proteins in the brains of these patients. So these little pictures you see here are the typical protein aggregates that neuropathologists can see in the brains of people with these different diseases upon post-mortem examination. Now, these diseases are devastating, and we've studied them for many, many years now, decades, even longer in some cases. And this enabled us to acquire a huge amount of knowledge. So there's been many hypotheses that were tested in thousands of clinical trials. This data is from 2019, so there's even more trials performed until now, 4 years later. But at the time, there were already more than 2,000 trials performed. And they tested a number of hypotheses that were all looking very promising in the preclinical studies that were performed. The problem is that a lot of these hypotheses have been failing. So in Alzheimer's disease, for example, it seems that if the data holds up, maybe we have one success. And this is a new drug. These were results that were announced just a few months ago. And this is a drug that is a monoclonal antibody called lecanemab. So this is an antibody that seems to recognize the protein aggregates that accumulate in Alzheimer's disease and may do something. They may change disease progression. We still don't know for sure, but there's data suggesting that this might be the case. So this tells us that although we have many hypotheses, we still have limited understanding of what is going on. So we need to think outside the box, and we need to investigate the molecular mechanisms involved in all of these diseases to a greater extent, because maybe we don't know enough yet, so we can treat them as we would like. And what we know is that genetics explains only a minor fraction of all cases of neurodegenerative diseases, especially the sporadic ones, of course. So these are all famous people that I'm sure you recognize. Some of them are already passed away. Others are still alive. But we know that these are not genetic cases. And these people had different lives. So one was a former pope, a president of the United States, an actor, and a sports person. Well, actually, two actors and politicians. So these are famous, very powerful people, and they still develop this disease. So the question is, why is it that as we age, some of us will develop this disease and others apparently not? We know we are all different. Genetics tells us we are all different. We live in different environments. We are exposed to different different chemicals during our life, different foods, different styles of life. So we're all different. This is no surprise. And we know that most of these diseases, like Alzheimer's and Parkinson's, they are complex. So what does this mean? It means that unlike in single gene disorders, of which Huntington's disease and other polyglutamines are examples, in those diseases you have mutations, so variations in a single gene, and then this can be affected by the environment and by lifestyle factors, and also by other genes, but there's a major component of a single gene. Now, in complex disease, what you have is you have many different variants in different genes, and alone they have a small effect. But then together with the environment, they can increase our risk for developing a certain disease. And this is true in a variety of diseases, in cancer, metabolic diseases, neuropsychiatric diseases, and neurodegenerative diseases as well. So we know now that there is this intimate connection between the genes and environment, and that there's an additional layer of complexity that is brought about by epigenetics. And here, I'm sure you heard about mechanisms such as DNA methylation, histone acetylation, also microRNAs and other types of non-coding RNAs. All of these factors can interfere with the way our genes are expressed and proteins are produced. And so this contributes to different aspects of these diseases. Now let's focus on the most common synucleinopathy. This is Parkinson's disease. So Parkinson's is a very complex disease, a very complex problem. And this is a slide that represents the progression of the disease. And here I show you the brain region that is affected in Parkinson's disease. So this is a region in the midbrain called substantia nigra, because as you see here in a healthy brain, there is this dark pigmented area that gives the name to the region, the substantia nigra. And in a Parkinson's person, you see that this region is not as dark. So the pigment, the neuromelanin that used to be in that brain region is gone. And what we know is that those neurons in the substantia nigra and in other brain regions accumulate protein aggregates, and these are called Lewy bodies. Now, during disease progression, these patients suffer, they're known to suffer from motor alterations that are shown here in this yellow box. And this is really what causes the patients to seek the neurologist, is when they have the motor problems. But now we know that even years before the onset of the motor problems, there are already other problems ongoing, such as REM sleep behavior disorder, constipation, depression, hyposmia, so the loss of smell that is also common in people with COVID-19. This is a sign of Parkinson's. And alone, these symptoms are not enough for one to say, oh, if you have hyposmia or depression or constipation, you already have Parkinson's disease. These are not specific, but they happen before the onset of the motor symptoms. Now, the genetics in Parkinson's, as I told you, is complex. And this is a schematic showing you that there's some single gene mutations that are very rare, and they have a very strong effect size, and so they cause Parkinson's. And then there's a large number of variants in many genes that are much more common, and alone, each of these variants does not cause Parkinson's, but together with the environment and in the context of multiple variants, they can cause or at least increase the risk for Parkinson's. So we need to understand all this molecular complexity so that we know exactly what is going on and why disease starts. But Parkinson's may have a problem of definition. So at this point, we don't talk about just Parkinson's disease. We should talk about Parkinson's syndrome. It's like in cancer. You know, if you say cancer, you don't know what type of cancer one is talking about. It could be breast cancer, lung cancer, pancreatic cancer, you know, any type of cancer. And if you say Parkinson's disease, this may also not be enough to tell us what type of Parkinson's disease it is. So we need to be better at defining this riddle. And here I created a small riddle just to illustrate what I mean. So just because something looks like it could be Parkinson's, it doesn't tell us enough. So it could be more than, or it could be a specific type of Parkinson's. And so the question is whether there is one disease called Parkinson's or whether there is more than one disease. And this is important because maybe we will treat these different forms differently. So we need to be able to distinguish them so that we can treat them in the right way. And I've been involved together with other colleagues in a task force of the Movement Disorder Society that I definitely recommend you check out. You, as students, you can become members without paying fees, and you have access to really amazing resources through. If you're interested in movement disorders, I recommend you look at this. But anyway, this society enabled us to put together a task force that is now working on the biological definition of Parkinson's. So the idea was to try to bring together information from neuropathology, clinical information, from biology, and come up with a better way for defining the disease to help clinicians and to help basic scientists know what we're talking about and how we may be able to treat this disease better. And so when you think about all of these aspects that we know are changing in Parkinson's, this shows you how complex it is. So we know that we have loss of substantia nigra neurons and Lewy body pathology. We know that there's imaging techniques like DAT scan or SPECT, maybe even PET, that can give us signals in the brain showing that there is a pathological alteration. One can measure also using MRI volume alterations of some brain regions. Then there's the genetic aspect that I already mentioned, and the motor aspects that are typical of Parkinson's. So it's very complex, and we need to really see the whole picture in order to understand what it is. And when we think about the possible molecular mechanisms in Parkinson's, the picture is also very complicated. So now we are zooming in on the molecular mechanisms, and we know there could be mitochondrial alterations, inflammatory alterations, and there's this component, the alpha-synuclein, here on the left side, alpha-synuclein aggregation. This is very important. And this result that I show you here, this tells us that alpha-synuclein really is probably playing a role. And this study that was done already 20 years ago showed that there are families of people where there is a duplication or triplication of the gene that encodes for alpha-synuclein. So these people have, because they have a higher number of copies of the gene, they produce more alpha-synuclein. So we know that if you produce too much alpha-synuclein, this is not good for your neurons in the brain. So I will focus now in the rest of my talk on this protein, on how we are studying this protein. And that's why in the title of my presentation, you saw synucleinopathies. So these are diseases where alpha-synuclein is aggregating in the brain. And how do we think about this? So for this, we need to think about very basic aspects of disease, and these have to do with cells. So this is a picture or a schematic of a typical animal cell. And I'm sure in your studies, you already saw many variations of this type of picture in different books or in the internet. And I'm sure you could all tell me what each of these compartments and organelles are, right? I think this would be easy for you. And you see here this space in between all these organelles. This is the problem of this schematic. This doesn't really capture the complexity of the intracellular environment. So we think that the intracellular environment is more like this. So there's no space between molecules, between proteins and sugars and lipids and nucleic acids. All of this is inside the cell, and everything has to work perfectly so that the cell can function. So it's really a big challenge, as you can imagine. So imagine in the cell, some proteins need to find other proteins. So it's like finding Wally, right? You all know these books, and I show you Wally's over there, so if you know where he is, you don't need to look anymore. But this is what the cells need to do when they need to find proteins that start to aggregate. So for this, the cells have what we call a proteostasis network. So this is a network of pathways that are involved in recognizing proteins as they start to misfold, and that try to deal with them either by recycling their components or sending other proteins to help refold and keep the proteins active and functional. And in the field of synucleinopathies, this German neuropathologist was very important. So in 1912, more than 100 years ago, he was imaging brains of patients with Parkinson's, and he saw some structures that he thought were interesting and were present in the brains of the patients. And he drew them, and you can see some of the drawings he made here. So this was the beginning of Lewy bodies. So he drew these structures, and then there was a Russian scientist that in 1912 saw these structures in the substantia nigra in the brains of patients with Parkinson's. So he called them Lewy bodies because it was this German scientist that saw them first, and then the Russian scientists called them Lewy bodies afterwards. So the way we think about these Lewy bodies is that they form through the aggregation of the protein alpha-synuclein. So the protein starts from a monomeric state, and then it starts to aggregate and form what we call protofibrils and fibrils, and eventually these fibrils come together in Lewy bodies. And they are made of this protein alpha-synuclein that I mentioned. This is not a very large protein. It has 140 amino acids. It's present in the presynaptic compartment, but also in the nucleus of some cells. And it's also abundant, not just in the brain, but also in the blood, in red blood cells. But we don't know what it does in the blood. So it could be important, but we study this protein much more in the brain than in the blood. And the function of the protein seems to be that it interacts with membranes with a certain curvature, and it goes from a state where only one part of the protein is connected to the membrane or other parts of the protein. So it seems that this connection to membranes is important for its aggregation. So this has come from the work of many colleagues in the field, some of them here from Gottingen and many from other places in the world. And to study this protein in the laboratory, we use models, and we use a variety of models, very simple cells like yeast cells, more complex animals like flies, Drosophila, or mouse models. And the way we think about models is that they are all wrong. They are only models. They are not humans. But if you ask the right question with the right model, then you may get important answers. So this is how we think about the models we use. And I'll show you how we are trying to find aggregated proteins in the cell and how we study them. So we develop different models, cell-based models, to study different aspects of the aggregation of alpha-synuclein. So to study this late stage aggregation product, we use cell models where we can produce the alpha-synuclein, and then using microscopy, we can see these inclusions that you can see down here, and they have many properties that look like those of Lewy bodies. So using these cell models, I show you here, there's cells that don't have inclusions, cells with large inclusions in a small number, and cells with a larger number of small inclusions. So we can count them under the microscope, and then we can study, for example, the effect of different mutations in this protein to see how they change the aggregation pattern. And you can see here, this graph with a lot of data shows that some increase the number of cells with inclusions, other decrease. So they have different effects, and we are studying all of this to understand how the mutations affect aggregation. But we want to study the types of aggregates that are not so easily to see with a microscope. So, to study the stars, what do you use? You use telescopes. To study certain animals, you don't need to use any imaging instrument, because you can just see the animals, and you can do a lot of studies without the need for any instrument. So you can just use your naked eye. To study cells, you need microscopes, because you need to see objects that are small. But to study proteins, you need not microscopes, but nanoscopes. So these are instruments that allow you to go in the nanometer range and see objects that are too small for normal microscopes to see. So we talk about nanoscopes or super resolution microscopes. And to study then these species that are small, these dimers and oligomers, we used another model that we call, so those larger ones you can see, but these smaller ones, these are the ones we want to see. And we use a model called bimolecular fluorescence complementation. So basically what you do is you take your protein of interest, in this case alpha-synuclein, you confuse it to fragments of a fluorescent protein, and then if your protein of interest interacts, then it forms these fluorescent complexes that now you can see. And these are only dimers and oligomers of your protein. So you can see species that you could not distinguish without this technique before. And again, using a panel of different mutations, we are looking at the effect of these mutations on the oligomerization. So I will not go into detail here. This is just to illustrate that we can use these models to image species that we could not see otherwise. So let's talk about this super resolution microscopy. In Gottingen, one of the recent Nobel Prize winners was this colleague called Stefan Hell. He got the Nobel Prize in chemistry in 2014. And what he did was he came up with a mathematical way for overcoming the limit of resolution of a microscope, which is around 200 to 150 nanometers. So by adding a component to the equation, he was able to extend this famous equation from Ernst Abbe that tells you that the limit of resolution of a light microscope depends on the wavelength of the light and the numerical aperture of the objective of the microscope. And using this approach that Stefan Hell invented, you can now resolve objects that are closer than what you could see with a regular light microscope. So now you can image objects that are as close as 50 nanometers apart. And this is a comparison of what an image looks like from a confocal microscope or a STED microscope. This is the name of the technique. It's a type of super resolution microscopy, and this is called STED. So you can see that now you can see the objects in much greater definition. So using this technique, we are imaging alpha-synuclein, and you can see here that inside the cells, you see species when you zoom in that are much smaller, and without this type of technique, you could not detect them using just normal light microscopy. Another type of super resolution microscopy we're using is this expansion microscopy. And basically here, what you do is you physically expand your sample. So you take a tiny cell, you make it swell, and now you have all the objects inside the cell separated through the polymerization of a gel that you can see here. So the gel starts small, and then by adding water, you can make the gel grow, and all of the samples that are embedded in the gel. So it's like the matrix in a diaper of a baby. There's a matrix, and when the baby pees, then the matrix absorbs water and expands, but the water, the liquid, is in the matrix, so it's not spreading and making the baby wet. So this is the same idea in expansion microscopy. And I show you here some examples of the results we are obtaining. So pre-expansion, so you see this is a normal image, and you see in blue the nucleus of a cell, and this is post-expansion. So compare the size of this nucleus with the size of that nucleus. So this is several times larger. And this means that all the objects inside a cell expand. And now you can see objects here in red, for example, the alpha-synuclein that you could not see before. And you can zoom in and see all this detail that before you could not resolve before the expansion. And you can do quantifications, you can count the number of assemblies in cultured cells, and you can see how it changes in different cell models. You can see all this detail. And here just some more examples of post-expansion, you see all these inclusions. And if you zoom in, you can start to see the details of the molecular architecture of these inclusions that look like ring-like inclusions inside the cells. But okay, so we can study the protein, we can see it, but we still have these questions. Why does the protein become toxic, and how does it cause synucleinopathies? How does it go from a normally folded structure into these aggregates that contain the protein? And we need to think here that proteins are more than just what is encoded by the gene, more than the amino acid sequence. And what I mean here is that proteins undergo a series of chemical modifications called post-translational modifications. And these are important because they change the folding, the localization, and the function of proteins. So you can think about any protein, your favorite protein, in my case, it's alpha-synuclein, but you can think about any protein, the linear sequence of amino acids. We know it undergoes these chemical modifications in different parts of the proteins. And what we think is that maybe at some point there's a combination of modifications that comes together and changes the conformation of the protein. And in some cases, this can trigger the aggregation. And these modifications, in many cases, they are useful, but in some cases, in some proteins, they could cause problems. But these are tools that nature evolved during millions of years of evolution to increase the complexity of the proteome. So we know in our genome, we have 25,000 genes. And then through alternative splicing and through post-translational modifications, we can significantly increase the complexity of the proteome so that there are all these different species that can perform different functions in our cells. And we think that maybe these post-translational modifications can explain the complexity in synucleinopathies. There are diseases that are characterized by the accumulation of alpha-synuclein deposits. So as I told you, I already mentioned Parkinson's disease, but there's other diseases like dementia with Lewy bodies and multiple system atrophy, where different types of alpha-synuclein inclusions accumulate in the brains of the patients. And we think that maybe these different aggregates form due to the occurrence of different sets of post-translation modifications in alpha-synuclein. So the way to think about this is that perhaps you start with the same protein, alpha-synuclein, and then through different post-translation modifications, you end up having slightly different shapes or structures in your aggregates, and this could be with a molecular explanation of the disease. This is something we and many groups are studying at the moment. And we know that this protein can indeed be post-translationally modified. We know in Lewy bodies in the brains of the patients, this modification called phosphorylation on amino acid 129. It's a serine, so it can be phosphorylated there. On serine 87, you can also detect phosphorylation. And so it seems that there's different phosphorylation sites, but there's many other modifications that can take place. And the idea now is that we try to zoom in and look at the whole sequence of the protein to see where the modifications are located. And then we study what they do to the aggregation of the protein. And in this context, we've been studying this connection between diabetes and Parkinson's disease. We know that the risk for Parkinson's disease increases in patients with type 2 diabetes. So there has been a recent study confirming that. And so we just don't know what is the molecular connection, but we think this may have to do with this type of modification called glycation. This is the non-enzymatic reaction of proteins with sugars, and this is what happens. So glucose as a sugar can react with certain amino acids in the proteins and form covalent bonds that eventually will damage the proteins and cause them to misbehave and to aggregate. So this is actually a series of complex reactions that we call leading to the formation of what we call advanced glycation end products. And so I will not show you data about this, but we want to understand this connection between diabetes and Parkinson's. But so just to end, if we think about this question, and we think alpha-synuclein is important, but the question is, are the Lewy bodies really causing the problems in Parkinson's disease? So are they good targets for therapeutic intervention? And this we cannot answer yet. So we do not know. The clinical trials performed so far have not been so encouraging, but maybe we just need to keep looking. We need to keep understanding what is going on. And for this, we need new scientists, new ideas, and we need all of you that are interested in this field to come and work and help us think about the problem, bring in new approaches, because we just don't know enough. Where we are today is that we know that the clinical symptoms that are typical in Parkinson's disease that have to do with alterations in the signaling and the circuitry in the midbrain and the cortex, this is what causes the movement problems, the motor problems. And today we have therapies that can treat some of these symptoms, especially levodopa. Maybe you heard of levodopa. This is the drug that is used in Parkinson's patients, but it treats only the tip of the iceberg. It treats only the clinical symptoms. So we don't know the causes very well. We have ideas. Maybe it has to do with protein aggregation or mitochondrial dysfunction or autophagy dysfunction, any of the molecular mechanisms that could be related, but we don't have therapies that can target these molecular alterations. And this is where we need a lot of research. So we need to understand the mechanisms so that we can develop future therapies for these diseases. One problem we have is that each of us can only study one part of the problem. So I study a little part, another colleague studies another part, but we need to distance ourselves so that we can see the big picture. This is the challenge. It's very complex, and we need to distance ourselves to really see the big picture. So, as I said, we need new people to study additional parts of the problem that then we can work together to try to solve it. So finally, just to thank some of the people in my lab, I actually have a student that is from Ukraine originally. She's also Portuguese, so she speaks Portuguese and Ukrainian, and she has been a PhD student in my lab for a few years now. And as you can see, it's a very international environment. People from India, from Portugal, from Chile, from Italy, from Egypt, from Croatia, from many different countries. This is a big advantage of research. We can work together with people from anywhere in the world. And if you're interested in studying alpha-synuclein, I recommend that you look at this special issue in the Journal of Neurochemistry. This was an issue that I edited together with some colleagues, and many of the major names in the field contributed to this issue. So you see articles by a lot of people in the field, so you can study different aspects of synucleinopathy. So visit this special issue and you will see a lot of useful information. And finally, I know it may not be so easy, but if you have a chance, there's really good conferences happening this year. I selected some that will take place in my home country in Portugal. One is now in May. It's this FANS regional meeting. It's a neuroscience meeting. Then there's this one in August 8 to 11 in Porto, my hometown in Portugal. And this is the International Society for Neurochemistry meeting. You can apply for travel grants and you can go there. And then there's also the prion meeting. This is one that I'm organizing. It's on prion disease and prion-like disease. So this is much more specific. This will happen in October. So if you're interested in any of these diseases, you should look them up online and try to register. And it would be great to see you. So I'll stop here. And if you have any questions, feel free to get in touch. Thank you very much.
So thank you, Professor, for such an interesting and breathtaking lecture. It was extremely interesting for me. I'm sure that it was interesting for our viewers as well. And now I would like to ask you a couple questions, if that's okay.
Sure.
Sure. So the first question is about the epidemiology of this pathology. So can we see an increase in the number of those pathologies worldwide or in some countries specifically?
Yes, so that's a very good question. So indeed, overall, we are seeing an increase in the number of cases. And why is this? Well, the reason is that fortunately, we are living longer. So there's more people reaching older ages due to change in lifestyle. And so some of these conditions are age associated. So it means that as we age, at some point, the brain cells don't work as well, and then we develop the disease. So because there's more older people in the world, then there's a larger number of cases. And so that's why the numbers are increasing.
The second question is just out of pure interest. It's a question for you personally. I've seen the picture of the iceberg and the treatment, and I guess this question may sound like just pointing your finger into the sky, but how many years would it take to have some effective treatment for those pathologies? What do you think?
Yeah. So here, of course, I can only speculate. It could be as quick as a year. We know that there's many clinical trials ongoing, and there is a lot of hope that some of these trials will be positive, and so they will show that we have therapies that work. But it could also take 5 years or 10 years. The reality is we don't know. We cannot predict. We know we are making progress. We know we are testing hypotheses, we have better tools, we have better models, and so we know we are going in the right direction, but it may be that we will not, maybe the next big findings will not be immediately a cure, but what we can expect is that they will start to change the progression of the disease, and this will be signs that we are going in the right direction. And then we may need to do combination therapies like we did for HIV, for AIDS. So it's not just one molecule, it's a combination of molecules that really has a strong effect. And here I think we should expect the same, that maybe individually we'll find molecules and drugs that have a small effect, and then as we start to combine them, maybe we'll have the greater benefit. So realistically, I think we may be a few years away, but maybe we'll start to see signs of effect in the coming year or two. That would be great. So that's my prediction.
Well, that sounds super interesting, and I'm sure that a lot of people who will watch this lecture will get more interested in this topic, and maybe we will have more researchers working on it in the nearest future. Thanks a lot, Professor. I want to thank you again for agreeing to do this lecture for us. And we hope to see you with us in the future as well, BIMCO 2024. So yeah, thanks a lot.
Thank you. All the best and stay safe, everyone.